Dear students, in today's lecture we will discuss about the different techniques for protein purification. First technique for protein purification is the chromatography. That chromatography includes basically two different types of columns. That column includes the pack column and wash column. The pack column is basically the column which is packed with materials which include resins that basically can absorb molecules based on different properties. That property could be size, property could be the charge, or that property could be the binding affinity of the proteins of which we want to separate. So second type of column is the wash columns that will in that column molecules are washed through the columns with the buffer. And third is the collection collect fractions, and then fractions are taken at some points your molecules will be eluted. So here after the purification of the proteins, the fraction will be effluents and that will be eluted from the here. So a second type of chromatography is the ion exchange chromatography. That chromatography basically this separates the protein on the basis of charge. There are elute proteins that increase salt or pH to elute the proteins of interest. Here the polymer beads which are present, which are negatively charged fractional groups which are present within the this column. Then protein mixture is added to the column that contains cation exchanges. So that polymers which beads which are basically the negatively charged, that will attract the proteins, and then these proteins will move through these columns at rates that is determined by the net charges at the pH which is being used. With cation exchangers, proteins with a more negative net charge move faster and elute earlier. So here is basically the protein is separated on the basis of charge. So that's that type of chromatography is known as the ion exchange chromatography. Next is the size exclusion or gel filtration chromatography. As the name indicates, here the proteins are separated on the basis of their size. As washed with the buffer, small molecules which will enter within the beads, so that these when the small molecules will enter within the beads, so that cannot freely move through these mixtures. So protein mixture is added to the column that contains cross-linked polymers, and the polar protein molecules which are bigger in size and larger molecules that will pass more freely, appearing in earlier fractions, while the smaller molecules will attach within these porous polymer beads and will exclude or will separate later. So in this size exclusion or gel filtration chromatography, basically the proteins are separated on the basis of their size. Next is the affinity chromatography. In this case, protein mixture is added to the column that contains the polymer bound ligand specific for the proteins of interest. Here the protein of interest, as I have discussed, that protein of interest is like moon like shape and ligands are like this black color. So when the mixture of protein is added, the protein of interest which will be of this type, that will attach with the ligands and that will be separated first, and then unwanted proteins are washed through wash columns, while the protein of interest that will elute or that will be attached with the ligand, and then that will be eluted by the ligand solution. Next is protein purification technique is the electrophoresis. In this the proteins are separated or here in the using the electrophoresis technique you can also separate the nucleic acid, etc., and that could be separated also by using the size or by the shape and the protein and the charges. Protein migrates based on their charge to mass ratios, and proteins are visualized using the radioactivity or using staining techniques, and then use the gels made of cross-linked polymers. Here you will add or you will run your sample through the gel, and then that sample or the proteins will be separated on the basis of their charge or on the basis of the size on the gel, and then this gel is read onward. Next is the isoelectric focusing gel electrophoresis. In this case, that is determined the isoelectric point of the protein, and then the proteins are separated until they reach the pH that matches their isoelectric point. So a stable pH gradient is established in the gel after application of an electric field, and then sample is loaded over here, and then after protein solution is added and electric field is reapplied, and then after staining the proteins are shown to be distributed along the pH gradient according to their isoelectric focusing point. Next type is the centrifugation. Here the proteins are separated by the size or by the density. Differential centrifugation. In case of differential centrifugations, these are separated large samples from the small particles. Here tissues are tissue homogenization is done, and then low speed centrifugation is applied. Then supernatants are subjected to medium speed centrifugation. Pellet contains whole cell nuclei, cytoskeleton, plasma membranes, and that pellet is removed, and then supernatant is further subjected for the centrifugation. Then after centrifugation for 20 minutes, then supernatant is again subjected to high speed centrifugation, and after again then pellet is removed, and again supernatant is collected, then subjected for the very high speed centrifugation, and then supernatant which contains the soluble proteins. That protein is of our interest. Next is the isopycnic or sucrose density centrifugation. Here centrifugation in sample is in isopycnic centrifugation, sample is centrifuged, then sucrose gradient is applied, and less density components will be placed downward, and more dense components will be down. These are the more dense components, and these are the less dense components, and then these are separated on the basis of their fractionation in different on the basis of their density. Then these are separated, these samples are separated. Thank you.